Okay, we are back again in a TIBSC Zoology Mammalian Histology paper number 2 semester 1 uh, class. Here we are discussing about the salivary gland from the syllabus 3.5 topic, that is glands associated with the digestive system. We have already seen the basic histology of glands, basic histology of different types of glands. Then we have seen the other about the salivary gland. Now different types and different structures and comparisons among the histology among the salivary glands. Now we'll see in detail every parotid, submandibular and sublingual glands. So here we have this parotid glands section. And interlobular connective tissue septa is shown, and interlobular duct. Now, among these all labels, this serous acini. When we say serous acini, I think now you remember what is that serous acini is. All that grape-like structures line internally with the epithelial tissue, and they are producing the serous. And with the characteristics of that serous, you see these cells are stained with a dark pink color. Okay, as it is a secretory gland, along with the tubule system, duct system and acini, what else you will see here is the lymphatic ducts and blood vessels. Okay, and all these lobules, these single lobules are separated with a layer of a connective tissue. So that connective tissue are interlobular connective tissue. They form the septums, a compartment, and the lobules are arranged. Here in this connective tissue portion only you will find that there goes this interlobular duct, and in this salivary gland you will see some accumulations of a fat accumulations represent in the form of a adipose tissue. Adipose tissues the present it represent in the form of a vacuoles, because that fat get dissolved in a process of staining microtechnique. So in a final section, when you observe under microscope, it looks aware empty spaces that represent the adipose tissues. Okay. So as we advance, we'll see different sections of parotid, submandibular and sublingual glands. What you have to do is to try to train your eyes to separate the identification of whether this kind of a secretory gland is a serous, mucus or mix. That is the critical point in this salivary gland histology slide identifications. Rest thing goes on same. They have all the glands, salivary gland, they have connective tissue septa, all the salivary glands have interlobular duct, interlobular, intralobular duct, but only the difference you will find is in these different types of serous acini, mucous acini and mixed acini. So here is a section of a submandibular gland now. What change you see here? The staining color change. Now here these are the acini, but do you see a dark pink color structure here? No, they are lightly colored structures and their nucleus shape is lantern and arranged at the periphery. So on that basis, we can say that this is not a serous gland and it is a mucous gland. So here you see that label mucous acini. But what about this serous demyelinus? You can see a dark color surrounding this acini, some dark color structure here. This dark color structure represent the serous cells, serous glands arranged on the periphery of this mucous acini. So we can say that this is in submandibular gland. What type of secretory glands are there? Mixed type of secretory glands are there. Mixture of serous as well as mucous acini. Serous as well as mucous cells you will see here. So it is a mixed type of secretory cells, glands you see in a salivary gland in case of a submandibular gland. Okay. What about these dark color structures? These are the pure serous acini. These are pure serous acini. What about here? These are the pure mucus acini. Serous acini separately mucus acini, and here mucus plus serous acini together. So mixed glands, secretory glands are seen in a submandibular section. Rest of the thing like interlobular duct is present there. The connective tissue septum is there, connective tissue forming the lobule and the septa, and that separate the lobules. Then here the large excretory duct also seen here, the major duct through which that secretion is poured through the submandibular duct into the mouth. And again here these small tubules represent the intercalated interlobular ducts. In this intralobular duct, you can see a small round structure here, intralobular duct. These are the interlobular, and these are intralobular duct. Then this serous acini, mucus acini, and serous demulans, that is a mix acini. Sublingual gland now. So here you have to see which type of acini are there. You forget about the connective tissue forming the septum. You forget about the blood vessels, presence of blood vessels. You forget, you remove from your view all that intercalated, interlobular and intralobular duct. What else remain is only the acini. You focus only on the acini here. Now, which type of acini you see here? They are very light in color, are lightly stained, nucleus is flat, and no dark color cells lying around the acini. So, which type of acini this is? So, mucus acini, purely a mucus acini is there. So, all sublingual glands are full of mucus acini, sublingual mucus secretions, submandibular mucus plus serous secretion, and earlier parotid glands totally serous secretions. Got it? Okay. Now just take a look and see where this is what these hollow vacuole structures are. What these are the adipocytes. Remember, just recall, these are the empty space. These are the vacuoles, empty vacuoles like structure, adipocytes, that fat depositions here. Then this structure, this is a interlobular duct or a duct which is collecting secretions and pouring on the surface. All these are the mucus acini, intralobular duct. Here smaller are intercalated duct and their interlobular duct. Okay, here is an actual color photograph of a section to reveal how that color patterns help you to identify. Now see this light pink color structure as compared to the dark color structure here. So all these dark brownish color structures stains all direct dark brown color structure stained here. These are acinis, but they are serous acini, and light color pink color structures acinis are mucus acini. One more point you can verify here is there is a lumen in the center of this acini, which is not present in the serous acini. What about this striated duct? This is a second level duct in a that interlobular duct connect together to form this striated duct, and this is a cuboidal epithelial lining is there, but you don't see any serous deposition or mucous deposition here. So it is a striated tube. Now this empty hollow space present here that represent the fat cells, adipose tissues, which get washed out during the microtechnic procedure. So these are the fat cells, faces or adipocytes. Now in this another slide again, here to make you acquaint with the color pattern and how this say the mixed kind of salivary gland cells you can able to identify. Now see here there is a very light color staining is there, and on the periphery of this portion there is some dark color cells are there. So all these dark color cells are serous cells and called as a serous demilins because they are lining over the mucus acini. This is the central portion of the mucus acini, and all this is the serous cells lining surrounding. So can you tell this section is from which gland, whether parotid, submandibular or sublingual, mixed submandibular gland? Okay, so this is a section of a submandibular gland presenting the mixed type of acini and mixed type of a gland, presence of serous demilins. Confirm that this is the submandibular gland section. Okay, here we'll revise some point and to check what you understood. We take a very small quiz here, MCQ. The first MCQ is all are major salivary gland except which one? Options are palatine, parotid, submandibular and sublingual. Major salivary glands are parotid, submandibular and sublingual. Minor salivary gland is a palatine. So palatine is the answer here. Second question is holocrine type of secretion is a feature of which gland here? Holocrine secretion. Options are mammary glands, parotid glands, sebaceous gland and pancreas. First, try to recall what is holocrine secretion. Holocrine secretion when whole entire cell get detached from the basal membrane and get released into the secretions, released into the cavity and pour outside. Entire cell get detached. That is the holocrine type secretions. Now that holocrine type secretion you will see in parotid gland. No, in a parotid gland we have all that serous secreting glands, and serous secretion is not holocrine type. It is a through exocytosis, merocrine secretions. Then sebaceous gland. In case of sebaceous gland also it is not holocrine secretions. In case of pancreas again it is not a holocrine secretion. So example, the correct answer is a mammary gland here. Sweat gland is an example of which type of gland here? Now based on their shape, simple tubular gland, sweat gland. Just remember, just try to recall from the scheme. Sweat gland there. Simple coiled tubular, simple branch tubular gland and simple alveolar gland. So I think that sweat gland is a simple coiled tubular gland. Here the next one is all are true about serous acini except there is only one condition is a false in case of this all related with the serous acini. They are smaller in size. Serous acini are smaller in size. They stain dark. Okay, they may present as demilins and wide lumen. Now, no doubt this condition is true for serous acini. They are small in size. This is again true that if they stain dark, here they also present the demilins, but their lumen is not wide because entire cell don't pour their secretion in a cavity, but the cell get pinched off to release their secretions. Okay, so this D condition is not fulfilled in this related to the serous acid. Expelling of secretion through contraction is a feature of which type of cell here? They release their secretions through the contraction. Serous cells, mucous cells, serous mammalians, or myoepithelial cells. Expelling of secretion through contraction. Contractions is a property related with myoepithelial cells. So wherever there is a myoepithelial cells, they can release their secretion by contractions. And more it is related with the mucous acini, which are supported with myoepithelial cells. They release their secretions. So here the answer is a myoepithelial cell. Okay, here we have some questions from a question bank of old university question papers. These are the questions related with this topic 3.5 glands associated with digestive system. These are the frequently asked questions from the previous year questions paper. Now here the 1 marks question generally they ask like give the names of salivary gland. Here you can write the name of major and minor glands. Then what is acinus? To describe the structure of acinus and the function of acinus and try to give examples. Then state the functions of sebaceous gland. The sebaceous gland is related with the skin structures where they produce sebum. But here I have included it because we are studying the glands topic. Their functions we have to produce that they produce the sebum. Then define goblet cells. This is again related with the mucus producing, but here I have included it. Goblet cells are the secretory cells, unicellular secretory glands. And they produce mucus. They are modified epithelial cells. Then what is the holocrine secretion? Just in some few earlier slide we have seen holocrine secretion when the entire cell get disrupted and release their secretions. What is an alveolar gland? If that this is based on the different shape of a gland, when the shape of a gland is a grape-like or round capsule-like structure internally lined with the epithelial cells, they release their secretion in the lumen and that secretion is poured outside. That is the alveolar kind of a gland. Give name of any one compound tubular alveolar gland. Now here you have to give the example of that tubular alveolar gland. If you remember, you can give the answer of that. There is a this mucus secreting glands are tubular alveolar glands here. What is the merocrine secretions? Here, in case of merocrine secretions, if you remember that mode of secretion, on the basis of mode of secretion, we have seen different types: holocrine, merocrine, apocrine secretions. In merocrine secretions, through the exocytosis, that vesicles release their content on the surface of a cell. That is the merocrine secretions. Now, long answer questions sometimes they ask as a give an account of histological structure of parotid gland. Now you have to describe the cross sections of parotid gland. In some you take a reference of some few earlier slide where I have shown you a section of a parotid gland. You try to draw diagram and explain all the structures you see in that. There you will see that serous producing acini, serous acini, you will see majority then intercalated ducts, interlobular ducts, everything you have to explain in that with the diagram. Describe histological structure of sublingual gland. Again, this color photograph I have shown you. You have to describe that sublingual glands where you will see majority of that mucus producing cells along with the line with the demilions. Some amount of demilin you will see line around that. mucus acini in that you have to show here. Describes the structure of submandibular salivary gland. Clearly, mixed kind of acinis you can show in this submandibular gland. You try to study the color photograph of that submandibular sections and describe every part you see in that. Then sometimes they ask short note on types of salivary gland. There you have to describe in short all three different types of salivary gland: parotid, sublingual, and submandibular gland. Then write short note on the parotid salivary glands. So same these questions, the answer of these questions, account of histology structure of parotid gland, you can repeat in the answer of parotid salivary gland short